So all are welcome to the classroom, and we are going to proceed from the inner content of the cell, that is the cytoplasmic content of the cell. And as you can see here that the fluid content of the cell, here look at the diagram, you can see the fluid content of the cell and here you can see several membrane bounded organelles are present, as well as the membraneless organelles are also present inside the cell that you learn about some briefly in your previous classes. You can see here that this is the mitochondria, this is one is the Golgi apparatus, and these are the endoplasmic reticulum, and this one is your mitochondria, and this one is your centriole, okay, and this one is your nucleus, and inside the nucleus you can see another dense structure, that is nucleolus, and this is the fluid content, okay. And if you consider that cytoplasm is your C, then these all particles are considered as the ships of the C of cytoplasm, okay. Now look at here, you can take the screenshot for your note purpose. And as we have already discussed that cytoplasm is the fluid content inside the plasma membrane of the cell, and plasma membrane is a thinner membrane which is selectively permeable in nature, and that cytoplasm contains many specialized organelles and also some chemicals inside it. And the function of cytoplasm is that it helps in exchange of materials between the cell organelle, and it acts as the store of many chemicals that I have already said, like your amino acids, glucose, vitamins, irons, and certain other enzymes, hormones inside it. So it is the site of certain metabolic pathway such as glycolysis. And as you know that metabolic process is the biochemical process in which both making of process, that means anabolism, and breaking down of process, that means catabolism process occurs, okay. And here the example is your glycolysis, and in your higher class you learn more about the glycolysis, and simply you have to learn here that the glycolysis is the breakdown of glucose, okay. Lysis means breakdown, so it is the catabolic process. Now look at here, then what we have discussed that your cytoplasm is consist of several organs inside it, and if you'll observe, several kinds of thin membranous structures are also present inside the cytoplasm, and they comprisingly forms the endomembrane system in the cell. And the endomembrane systems of the cytoplasm generally consist of four main organelles. These are endoplasmic reticulum, Golgi apparatus, lysosome, and vacuoles. You can see their location in the cell in this diagram. Then first we have to discuss about what is endoplasmic reticulum. And as you know that from the outer layer of the nucleus, as you know that the nucleus has two membrane, so from the outer layer some extension occurs, and that extension forms the cisternae, the membrane like structure. These are called as cisternae, okay, and they extended towards the cell membrane, okay, and this extended layer forms a network of cisternae which is called as your endoplasmic reticulum, and it forms two parts in the cell. This inner part is called as luminal compartment, and the outer part of the endoplasmic reticulum system is called as cytoplasmic compartment or extraluminal compartment. Then the endoplasmic reticulum are of two types. As you know, the endoplasmic reticulum which contains ribosomes upon its external layer, it is called as rough endoplasmic reticulum, and the part of endoplasmic reticulum which doesn't consist any ribosomes upon it is called as smooth endoplasmic reticulum, okay. And you know very well what is the function of ribosome. Ribosome's function is it helps in the protein synthesis. Now look at here, the smooth endoplasm. One information for you that the smooth endoplasmic reticulum of muscle cell is called as sarcoplasmic reticulum. Okay, you can take the screenshot. This one is one information for you all. Then we have to learn about another membrane system of the endomembrane system is Golgi apparatus, and that Golgi apparatus, and listen, these all are the functions of your endoplasmic reticulum. Listen, sorry, I have to discuss about what are the functions of endoplasmic reticulum. These are the some glimpse of the endoplasmic reticulum. It helps in detoxification of drugs. Generally in liver, the detoxification of drugs occurs. Okay, the endoplasmic reticulum of your liver. Then metabolism of carbohydrates occurs by the endoplasmic reticulum, and it also helps in synthesis, means formation and transports of proteins and lipids. Okay, and as you know that ribosomes helps in protein synthesis. That means the amino acids are arranged by the help of ribosomes by decoding the message of mRNA that we have already learned in our previous classes in case of a prokaryote. Now you can take the screenshot. Yes, you can take the screenshot for the functions of the structure and function of endoplasmic reticulum. Yes, Bhumi is made up of proteins and lipids, and this has been revealed by the biochemical analysis of the cell membrane, structure of cell membrane, and that proteins and lipids prepared by the endoplasmic reticulum gets engaged in the cell membrane. So cell membrane ultimately helps in membrane biogenesis. Okay, and you can take the screenshot for another part of your notes. Look at the first point. Some of these proteins and lipids help in building the cell membrane, and this process is known as membrane biogenesis. And some other proteins and lipids they act as the enzyme and hormones. So that's all about your endoplasmic reticulum. Now look at here. You can see that this part of the cell is saying itself that I am the ugliest organelle of the cell. Look at him. Okay, but as we have previously discussed that it is not the ugliest one. It behaves like your PT cell, and probably you can recall the organelle of your cell. What is it? It is the, yes, it is your Golgi apparatus. Okay, so this organelle has discovered by Camillo Golgi in 1897. Okay, look this look at here. Camillo Golgi has discovered another endomembrane system of organelle of endomembrane system, that is your Golgi apparatus, or it is called as Golgi complex, or simply you can call it as the Golgi or Golgi body, and in case of plant it is called as dictyosome. Okay, and this organelle has its main role in the endomembrane system, and as you know it has three functions. Majorly it helps in packaging, it helps in collection, packaging and distribution. Collection, you can see this Golgi apparatus helps in collection, packaging and distribution. And or you can say, instead of distribution you can write, your favorite teacher is your PT sir, you can write this. Okay, the main functions of your Golgi body is these three. Collection, C stands for collection, and for your mnemonics it will be better for that C your favorite PT sir. Instead of C you can write C your favorite PT sir. Okay, and look at here, and the main function of the Golgi body is these three. It helps in collection of material from the endoplasmic reticulum, and P stands for it helps in packaging of the collected materials, and T stands for transport of material. These are the three important functions of your Golgi apparatus. And we have to learn about the structure of Golgi apparatus. Look at here. Here you can see the structure of Golgi apparatus, and here you can see that the location of Golgi apparatus in the cell, and generally it remains nearer to the endoplasmic reticulum, and it has one face. Look at here, it has one face which is concave and aligns towards the endoplasmic reticulum, and this phase is called as cis phase, okay, or incoming phase. Look at here, here is the incoming transport vesicles which is going to enter through the cis phase, that means the collecting phase, okay, and these structures are called as cisternae. Tubular structures are called as cisternae, and the internal structure, internal zone is called as lumen of the cisternae, okay, and you can see its another phase is trans phase. That means through that phase the Golgi apparatus transports the materials outside from the Golgi body, and generally you can see here that, look at here, you can see here that from the endoplasmic reticulum the ribosomes prepare the material protein, and proteins are transported through the endoplasmic lumen, then it is coming outside and it is entering through the cis phase of the Golgi apparatus. Look at here, it is entering through the cis phase of the Golgi apparatus. Then in that Golgi apparatus it again gets packaging, okay, by changing by after modification that packaged material returns out from the trans phase of your Golgi apparatus. Then after it remains nearer to the cell membrane, or sometimes it comes out from the cell membrane, and that release process is called as exocytosis. If cell releases some material from the through the cell membrane, then it is called as exocytosis, and endocytosis means entering of some material. Look at here, endocytosis is another process through which cell intakes matter from the outer environment by forming vesicles from the cell membrane. You can see these are the vesicles, okay, and here you can see this one is the lysosome. This one is the lysosome, it contains the digestive enzyme, okay. So that's all about your function of your what Golgi apparatus, structure and function of your Golgi apparatus. You can take the screenshot for your better knowledge. Then now you can take the screenshot for a note purpose, okay. Singularly cisternae and plurally just cisterns. Now the next part of your endomembrane system is your lysosome, and as you know that lysosome contains hydrolytic digestive enzymes, and generally such enzymes are lipases, proteases, carbohydrates, and these organelle has the main role to keep clean of the cell, so that it is called as the cleaner of cell, and sometimes these lysosomes help in digesting the unwanted materials from the cell, so that they are called as the scavenger of the cell. And also when the cells is going to die, then at that time what happens, the lysosome bursts and the digestive enzyme itself digest all the materials present in the cell, so that lysosome is called as suicidal bag of the cell. And generally the hydrolytic enzymes are what are the present in the lysosome are lipases, proteases, carbohydrates. Lipases are the enzymes which helps in the breakdown of lipids, and proteases are the enzyme which helps in digestion of, breakdown means digestion of your protein, and carbohydrates are the enzymes which help in digestion of carbohydrates. And these and another type of nucleases are also another enzyme, they can also digest the nucleic acid in the cell, and these all enzymes are also present in the lysosome. You can take the screenshot for the note purpose. And the enzymes which are present inside the ribosome, it is prepared by the raw endoplasmic reticulum, okay. Now look at here, our next point of discussion is the vacuoles, which is the fourth organelle of your endomembrane system. Initially you have learned about endoplasmic reticulum, then Golgi apparatus, then lysosome. Now is our vacuoles. And look at here, the vacuoles contains nearly about 90% of volume in case of a plant cell, and the outer covering of vacuole is called as tonoplast, okay. And generally your vacuoles contains sap, water, excretory products. Sap means some nutrient material which are remains in the storage form in that vacuole. Okay, and some excretory material and unwanted substances are also remains inside the vacuole, and generally those materials moves against the concentration gradient. That means from the lower concentration to the higher concentration of certain material, they transport through the tonoplast. This membrane is called as tonoplast, through the tonoplast, and remains stored in the vacuole. See, look at here, this one is the position in which the materials are transporting through the tonoplast inside the vacuole, okay, from its lower concentration to the higher concentration. That means against concentration gradient. And again I am repeating, if the molecule will transport or diffuse from the higher concentration to the lower concentration, then that is called as along the concentration gradient. That transportation occurs along the concentration gradient. And if transports occurs from the lower concentration to the higher concentration, and that type of transportation is called as against the that transportation occurs against the concentration gradient. Okay, take a screenshot for your note purpose. Now look at here. Now remember the name of scientist is George Palade. In 1953, he discovered the protein factory of the cell. And as you know that, what is the protein factory? Ribosomes. And ribosomes are also very much famous because of its size. These are the smallest organelle of the cell, remember. And that observation made by George Palade to observe the ribosomes. Okay, now you can see the structure of ribosome. Generally the ribosome contains, look at here, the ribosomes contains RNA. This one is the structure of RNA, okay, mRNA, and the protein, certain kinds of protein. Here look at here, these are the proteins and these are another group of proteins, okay, and they simultaneously forms the ribosome. And you can see, and as you know that in case of prokaryotes, they consist of the 70S type of ribosome. In case of eukaryotes, they are made up of 80S type of ribosome. And as you know that A stands for Sage-Bott's unit, and that Sage-Bott's unit is the unit which is used to detect the volume, that means size and density of the ribosome. And that Sage-Bott's 80S unit is comprises of, that 80S type of ribosome in case of eukaryotes is comprised of 140S unit, 140S unit, and look at it, this one is 140S unit and this one is 160S unit. But in case of prokaryotes, look at it, 40 plus 60, it is 100. Another 20% of that 40S type remains embedded inside the 60S type. So that simultaneously the total shape is, or the volume or density of that ribosome is 80S. And in case of prokaryotes, look at here, they have the 70S type of ribosome. And one thing you have to remember, in case of mitochondria and plastids, they also contain ribosome that are 70S type of ribosome. Okay, look at here, in case of prokaryotic ribosome, they have the two units like your eukaryotic ribosome. One is 50S unit, another one is the 30S unit, and they simultaneously form the 80S. If you add these 50 and 30, you will get 80, but some part of that 30S unit remains embedded inside the 50S unit, and so that the volume changes into 70S. Okay, now look at here, and ribosomes generally decode the mRNA. That type of diagram you have already seen. Decode the mRNA in a, look at it, here is your ribosome, here is the ribosome, and here is the ribosome that getting detached from the mRNA. And the number of ribosomes when attached along with the mRNA, they form the polysome, polyribosome, or simply you can say the polysome. And they helps in the formation of protein by arranging the amino acid in a chain manner. You can see that these are the proteins molecules they are getting. These are the protein molecules, they are formed by the arrangement of certain amino acids by the help of ribosomes. And generally ribosomes decodes the message carried by the mRNA from the nucleolus of your nucleus. mRNA generally carries information from the nucleolus of your nucleus, and that message is decoded by the ribosomes, and ribosomes arranges the amino acids to form the chain of amino acid, which later on develops into proteins. Okay, because proteins are made up of amino acids. Now look at here, this is the ultra structure of your muscle. This one is the microscopic view of your muscle cell, and here you can see that it consists of number of, look at here, some red color structures at here, and these are the stained part of this muscle cell, and these are stained means colored part, okay, and these are known as your mitochondria, which is the powerhouse of the cell. And yesterday I got a new notice from the new information about the mitochondria. I'll say, listen. And as you know that mitochondria is your powerhouse of the cell, except that one interesting fact you have to learn, remember that the mitochondria has its own DNA that you have already learned, and that DNA generally, in case of animal beings, the DNA generally comes from the mother, which are present in the mitochondria. That means mitochondrial DNA generally comes from the mother. Listen, during the course of fertilization, what happens? The wait the egg of the mother, has its own nucleus, and the sperm has its tail part, and the nucleus remains in the head, and the in case of at this side the sperm has its mitochondria, but in the cytoplasmic part of the egg cell has its mitochondria. And when sperm enters inside the egg, then only the head parts remains inside the head, and rest of the part cut out and they remains outside of the cell. So ultimately the mitochondrial present at the tail region of the sperm, they remains outside of that fertilized cell or your zygote, so that all the mitochondrial DNA which are present in the zygotic cell, they are the DNA that comes from the egg cell. So ultimately the DNA present in mitochondria migrates from the or mutates from the mother cell. Okay, this one is the information. Now listen, the length of that cylindrical structured mitochondria is about 1 to 4.1 microns, whereas its diameter generally ranges from 0.1 to 1 micron. Remember that diameter ranges from 0.1 to 1 micron. Okay, this one is the diameter. Okay, so this one is the size of your mitochondria. And as you know that mitochondria is a double membrane bounded structure. It has the outer membrane, look at here, this one is the outer membrane and this one is the inner membrane, but the inner membrane is not continuous. Inner membrane is discontinuous, and that discontinued part forms the finger like projections into the matrix region. This one is the matrix region of the mitochondria, and that finger like projections is called as crista. Okay, that finger like projection that enters into the matrix of the mitochondria, they forms the crista. And look at here, that yellow color layer is your inner membrane, okay, and that red color layer is your, or the brown color layer is your outer membrane. So they forms two compartment, one is outer compartment and another one is the inner compartment. Okay, that folding of the mitochondria, inner membrane of the mitochondria forms two compartment. And there are various types of enzymes that are present in the mitochondrial membrane. Look at here, you can take the screenshot, that the enzymes of outer membrane are monoamine oxidase, fatty acid coenzyme A ligase. Then enzymes of the inner membrane are ATP synthetase and ferrochelatase. And generally the outer membrane is selectively permeable nature and they allow certain material to enter in it, whereas the inner membrane forms the foldings which are called as crista, and it contains ATP synthetase enzyme which helps in ATP synthesis. And you can see inside the matrix of the mitochondria, it consists its own DNA, okay, and it consists its own ribosomes and RNA also. So it can produce its own protein, so that it is called as your semi-autonomous organelle, because to carry out all the functions of your ribosome partially depends upon the nuclear DNA. Since it contains its own DNA, it can produce its own protein, okay, so that it is called as semi-autonomous organelle of the cell. Except that ribosome also produce the ATP from its site of ATP synthesis, and here it contains the enzyme ATP synthetase. Okay, fine. Now take the screenshot for the structure and function of mitochondria, and it produces the energy in the form of ATP which is the energy currents of the cell, and ATP stands for adenosine triphosphate. Now comes towards our next component of your cell or organelle of the cell, that is your plastid. And plastids are of three types. One is chloroplast, chromoplast, and another one is the leucoplast. And here you can see the structure of your chloroplast. And chloroplast is another double membrane bounded organelle. And another thing you have to remember, the plastid is the largest organelle of your plant cell, whereas mitochondria is the largest organelle of your animal cell. Okay, because in case of animal cell there is no plastid. And look at here, here is the chloroplast, and chloroplast has two membrane. One outer membrane is the outer membrane is continuous, and the inner membrane is also continuous. But in case of mitochondria, their inner membrane is discontinuous and they forms the crista. Okay, and inside the inner membrane they have the fluid content that is called as stroma. That fluid content is called as your stroma. Okay, and inside the stroma these coin like structures are stocked upon each other, and each coin is called as thylakoid, and that whole stock is called as granum. It is called as granum, and its plural is grana. Okay, granum, its plural is grana. And each thylakoid membrane, look at here, each thylakoid is a coin like structure, okay, membranous structure, and each thylakoid has the chlorophyll, which is the green color pigment, is present, and they forms the photosystem, which helps in photosynthesis. Except that another structure of that stromal content is that, look at that yellow structure, it is called as stroma lamellae, which connects the one granum to the another granum. Okay, and some chemical reactions are also occurs during the course of photosynthesis at the stroma lamellae also. You can look, this one is the length of your mitochondria, which is generally the length of the plastid or your chloroplast is 5 to 10 millimeter, and its diameter generally remains in the range of nearly about 1 to 2 millimeter or 1 to 3 millimeter. 2 to 4 here, look at here. And so in case of some animals, is some kind of organism, you can see that whole organism is consist of one chloroplast. Look at here, here is your chlamydomonas, it consists of only one chloroplast. In case of algae, it is made up of number of chloroplast per cell. Okay, and in case of plant cell, the mesophyll of plant cell contains chloroplast, which provides the green color to the plants. And see what is the stromal content of the plastid. It generally contains the circular DNA molecule, which is double stranded in nature, and 70S type of ribosome in it, and also enzymes which are required for formation of carbohydrate and proteins in the chloroplast. That stroma contains all these required enzymes. And look at here, you can see the position of chloroplast in the cell. If you'll see, this one is the leaf, and you'll take this part of the leaf, it contains that type of cell. Okay, that mesophyll cell generally consists of chloroplast, and if you enlarge one chloroplast, you can see this one is the thylakoid, which is arranged in a stalk manner to form the granum, and granum's plural is grana. You can see this one is the plural form of granum. And that gramins remain floated inside the stroma, and each granum is connected with another granum by the help of stroma lamellae, and that thylakoid membrane contains chlorophyll color pigment, which helps in trapping the solar energy, and the whole chloroplast is covered by two membrane, one is outer membrane and another one is the inner membrane. That's all about your chloroplast. Then we have to come about the next. Look at here, here is the cluster of photosynthetic pigments present in the chloroplast. Okay, this one is the thylakoid membrane, and thylakoid membrane is also made up of, look at there, lipid bilayers are there. Okay, and look at here, you can see the grana. And then the next type of your plastid is chromoplast, and generally chromoplast consists of two words. One is chromo, means color, plast means plastid, and it provides different color to the plants, like it provides red color, yellow color and orange color to the plant. Okay, and here you can see that the another two types of pigments, like your fat soluble pigments except chlorophyll, those are the carotene and xanthophyll. Carotene and xanthophyll, they helps to provide such type of color by remaining inside the chromoplast. Okay, then another type of plastid is there, they are the leucoplast, and leucoplast generally helps in storage purpose. And if the leucoplast stores starch inside it, it is called as amyloplast. And if the leucoplast stores oil and fats inside it, it is called as elioplast. And if the leucoplast stores proteins inside it, it is called as aleuroplast. Then take the screenshot for the function of your plastids, structure and function of plastids. Now see the comparison between two types of chloroplasts, two semi-autonomous organelle of the cell, majorly generally in plant cell, that they both have their own DNA as well as ribosome and RNA, so that they can prepare their own protein. Hence they are called as semi-autonomous organelles of the cell. Very good, and thanks for joining the class.